Hi students, welcome to the WAPSI. Welcome to the next biology class. Here the first question. The enzyme recombinase is required at which stage of meiosis 1? You heard about meiosis, mitosis, etc. In the first year biology, there is the most important chapter, that is cell cycle and cell division. Cell cycle and cell division. It is more important because it is connected to many other chapters, such as the previous chapter, cells, the unit of life. Second year chapters such as principles and next genetics, molecular basis, etc. So in the chapter cell cycle and cell division, there are many stages of cell divisions. That is interphase. That means any cell cycle which contain, there are two stages, interphase and M phase. There are two stages of cell cycle, interphase and M phase. The term interphase, which means the stage in between two M phase, or the stage in between two mitosis. The term inter, which means in between. Interphase is the longest stages of cell cycle. Hence it is divided into three substages. They are sequentially, they are G1, S, then G2. Each stage has its own characteristic feature. So interphase, which is divided into three substages, they are named as G1, S, G2. Gap 1, synthesis, gap 2. Some cells, they are permanently or temporarily devoid the capacity to division and enter into another stage from G1 stage, which is known as G0. Which is G0 or Kcet stage. Not only all cells, some cells, they are enter into G0. After some time, they regain the capacity. If they regain the capacity for division, they enter into G1 and then through the S, G2, and they complete the cell cycle. Then the first stage is interphase, G1, S, G2. Then the next, second stage is M phase. Here, M phase, which is mitosis or meiosis. There are two types of division, mitotic cell division or mitosis and meiotic cell division or meiosis. These two are, compared to interphase, these two stages are complex stages. But which one is more complex? That is meiosis. Meiosis has many substages and it is complex than mitosis. Then these two are cell division process, but mitosis generally known as equational division. What is equational division? Meiosis is known as a reduction division. What is a reduction division? That means equational division and a reduction division. Why mitosis is known as a equational division? The characteristics of an equational division: if a parent cell which is diploid in nature and they undergo cell division and they produce two daughter cells, and these two daughter cells are also diploid in nature and structurally they are two equal halves of their mother cells and they contain equal, quantitatively and qualitatively they are equal. If a parent cell which is diploid in nature, they undergo cell division, it is said to be, if it is said to be equation division, they produce two daughter cells. If the parent cell is 2n, obviously the daughter cells are also 2 in nature, but they produce 2. After equation division, 2 daughter cells are formed. That is the main characteristic feature. But in a reduction division, if a diploid cell undergo reduction division, why it is a reduction? Here, there is reduction or reduces the chromosome number. In the case of mitosis, there is no change in the chromosome number, but in the meiotic division, there is a change in the chromosome number. There is a reduction in the number of chromosome. How much chromosome reduces? How much or how many chromosome reduces? Here, the chromosome number reduces by half. That means 2n reduces into n. Their diploid nature changed to haploid nature. Then, how many products are formed after meiotic division? There are 4 daughter cells are formed. Each daughter cells are haploid in nature and there are 4 daughter cells are formed. So, if a parent diploid cell undergo meiotic division, they produce 4 haploid daughter cells. So, there is a reduction in the chromosome number. So, the process is known as reduction division. Hence, meiotic division is a reduction division. So, the number of product, they are 4 daughter cells. And the major difference, another difference is mitotic division that happens as one process or one division or one stage. It is one mitotic divisional stage. But meiotic division, 2 stage, it happens by 2 stage. What are they? Meiosis 1 and meiosis 2. There are 2 stages, meiosis 1 and meiosis 2. Then, for mitosis and meiosis, whether it is mitosis or meiosis, the divisional process takes place by many substages. These substages are generally karyokinesis and cytokinesis. Karyokinesis and cytokinesis. Kinesis is the division, karyokinesis is the division of nucleus, cytokinesis is the division of cytoplasm or cell division. That means first karyokinesis takes place, after that cytokinesis. In the case of mitosis or meiosis, the first step is karyokinesis. So, the karyokinesis is the complex process than cytokinesis. So, however, it consists many substages also. Karyokinesis divided into 4 substages. They are prophase, the next one is metaphase, followed by anaphase, then telophase. That is the sequence of mitosis. What are the sequence of mitosis? Is prophase, metaphase, anaphase, telophase and cytokinesis. But in the case of meiosis, meiosis happens with the 2 stages, meiosis 1 and meiosis 2. The complete meiosis, meiosis 1 and meiosis 2, which involves meiosis 1 and meiosis 2. In the meiosis 1, there is a karyokinesis and a cytokinesis. That means karyokinesis is divided into prophase. So, the first prophase of meiosis 1 is prophase 1. Obviously, it is meiosis 1, which is divided into prophase 1, metaphase 1, anaphase 1, then telophase 1. After that, the cell enter into cytokinesis 1. It leads to the formation of 2 daughter cells. They are haploid in nature. A diploid cell which undergo meiotic 1 division and they produce 2 haploid daughter cells. So, the actual division takes place in meiosis 1. So, meiosis 1 is generally known as reduction division. Then, these 2 daughters, product of meiosis 1, they enter into meiosis 2. Before that, there is a short stage that is known as interkinesis. Interphase and interkinesis, they are 2 stages. Interphase that is the stage in between two mitosis. Interkinesis, it is a stage, a short stage in between meiosis 1 and meiosis 2. So after there is an interkinesis, the cell enter into meiosis 2. Obviously there is prophase 2, metaphase 2, anaphase 2, then telophase 2. After the cell enter into cytokinesis 2. Each haploid cell produce two daughter cells. They are haploid in nature. A haploid cell which produces haploid cells, two haploid cells. That is also equation division. So meiosis 2, it is almost similar to equation division. Meiosis 1, it is a reduction division. So this is meiosis. So meiosis is the most complex compared to meiosis. Mitosis, meiosis is the most complex process. Then our question is related to meiosis 1. The enzyme recombinase is required at which stage of meiosis 1? So among the substages of meiosis 1, prophase 1 is the longest stage. Longest stage of meiosis 1 is the prophase 1. Why this stage is longest one? There contain many other substages also. Prophase 1 again categorized into 5 substages. They are leptotene, zygotene, Kcet, diplotene, and diakinesis. Leptotene, zygotene, Kcet, diplotene, and diakinesis. Each stage has its own characteristic feature. Among these, zygotene, Kcet, etc. that is more important. Then the characteristics of leptotene. During this stage, the chromosome of these cells, they are thin fiber or hair-like structure. They appear as a thin fiber. That means condensation of chromosome just started. Chromatin reticulum condensed to form chromosome during the prophase. So that process started. Hence the chromosome there appear as thin fiber. So the stage is leptotene. And these chromosomes appeared in a bouquet formation or bouquet stage. So bouquet stage is the characteristics of leptotene. Then the next stage is zygotene. This stage is more important because some process that takes place during the zygotene. Zygote is the product, fusion product of male and female gamete. So zygotene, this stage, they have a similar character because zygotene is characterized with pairing or synapsis. What is synapsis? Synapsis is the pairing of non-sister chromatid of homologous chromosome. There are many terms: homologous chromosome, non-sister chromatid, then pairing. First, what is homologous chromosome? What is chromosome? And then we study homologous chromosome. Structurally, a chromosome which contain a central primary constriction which hold two cylindrical structure, they are known as chromatid. This chromatid actually condensed product of chromatin reticulum. So chromatin reticulum condensed to form chromatid. So a chromosome which contain two chromatid They are adjoined at a common point which is known as centromere. So this one is centromere, and these are chromatid. So a single chromosome having one, then two chromatid. Then the surface of the chromatid, there is a disc shaped structure which is known as kinetochore. Kinetochore. Not only every time chromosomes, or there is a kinetochore, there is chromatid, there is no, but some stages, that means these chromosomes are clearly visible. We have to see the centromere, kinetochore, chromatid, etc. So this one is a chromosome. Then what is homologous chromosome? Actually, homologous chromosomes are two chromosomes. Homologous chromosome, they are two chromosomes. They having homo, that means same. Locus, gene locus or location. Two chromosomes having same gene location, that is known as homologous chromosome. Our chromosome number is, what is the chromosome number of human being? Our chromosome number is 46 or 23 pair. Why it is known as 23 pair? Among these 46 chromosome, 23 chromosome coming from father and 23 chromosome coming from mother. And there is 46 chromosome, one father, one chromosome coming from mother. They are a pair of chromosome. Why these are considered as pair? Because they have a same gene location. That means one of the chromosome, or the first chromosome of the father and the first chromosome of the mother having the same gene location, they are known as homologous chromosome. So if this one is a chromosome, first chromosome derived from the father, so obviously the first chromosome derived from the mother, they are similar to the chromosome of father, that is there are two chromatid and they are joined at a common point which is known as centromere, and there is genes. That genes, this dot represented as gene. The first gene, second gene, third, fourth, fifth. This one first, second, third, fourth, fifth. Here also first, second, then third, fourth, fifth. First, second, third, fourth, fifth. That means this chromosome contain, the one chromatid of the chromosome contain 5 genes. And each gene expressed, or the expression of each gene, here the first gene, the height, that means tall. Obviously, here this one is tall. This one is height represented tall or dwarf. This one is tall. If it is tall, this one is also tall. If this one is dwarf, this one is also dwarf. Or small t. It is represented as small t. So these are two chromosomes having the same gene location, hence it is known as homologous chromosome. So the chromosome I draw here is a homologous chromosome. That is homologous chromosome. Then the non-sister chromatid. What are the sister chromatid and the non-sister chromatid? The chromatid of a single chromosome is known as sister chromatid, which contains same gene. That means the copy of one gene, the replica, photostat of one gene. Here this one is also sister chromatid. But this chromatid and this chromatid, that means the chromatids of two chromosomes are non-sister chromatids. That means the chromatids of two homologous, one chromatids of two homologous chromosomes are considered as non-sister chromatid. So pairing occur between non-sister chromatid of two chromosomes. What substance which adheres or which helps in the adhesion of or pairing of these two chromatids? That is known as synaptonymal complex. Synaptonymal complex is a protein which helps in the adhering of two chromatids or two chromosomes. This formation of a synaptonymal complex, that takes place in this zygotine stage. Synapsis, that is the pairing of non-sister chromatid of two homologous chromosomes, that takes place also in the zygotine stage. So zygotine is the characteristics with the synapsis. The next stage is Kcet. Kcet is the most important stages of meiosis 1 because meiosis, the function of meiosis 1 actually happening in the Kcet stage. So Kcet stage is characterized with the crossing over or recombination. What is recombination? What is the difference between recombination and pairing? Pairing or synapsis is the pairing of non-sister chromatid, but recombination is the exchange of genetic material. That means exchange of gene between non-sister chromatid of homologous chromosome. Exchange of genetic material between non-sister chromatid of homologous chromosome is known as a recombination. For that, there is a need of recombinase enzyme, such as restriction endonuclease enzyme, then ligase enzyme. These are the two major recombinase enzyme. What are they? Recombinase, restriction enzyme, nuclease, and ligase enzyme. So the formation of these enzyme that is happening or that takes place in the Kcet stage. So recombination is the most important stage. So that takes place in Kcet. Recombination helps in the variation, heredity and variation. The variation is due to recombination. Then diplotin. Diplotin is the stage with the helps in the chiasma formation. What is chiasma? Chiasma is the X-shaped structure. How it is formed? That X-shaped structure is formed due to the dissolution of synaptonymal complex except at the site of crossing over. Degeneration or dissolution of synaptonymal complex except at the site of crossing over leads to the formation of an X-shaped structure which is known as chiasma. Chiasma formation that takes place during diplotin. Then the next stage is diakinesis. Diakinesis is terminalization of chiasma. Terminalization. What is meant by terminalization? It is the movement of chiasma towards the tip. From the center to the tip. Here the movement of chiasma. Chiasma is an X-shaped structure. When it is moved up to the tip, there is no X-shaped structure. There is a V-shaped structure because their tip of the chromosome, they are adjoined at that time or joined at that time. Hence, it is not an X-shaped. From the tip, it is a V-shaped structure. So the movement of chiasma from the center or where it is formed towards the tip, this movement is known as terminalization. So terminalization takes place during this diakinesis. So the leptotin, zygotin, Kcet, diplotin, and diakinesis that takes place during meiosis 1. So meiosis 1 is the most important stage. Then the major difference between meiosis and mitosis. Meiosis and mitosis is in the mitotic division or mitosis, one diploid cell undergo a division and produce two diploid daughter cells. During these steps or during the process, there are change in the amount of chromosome also. That means 2n number of chromosome which bear 2c amount of chromosome. Then 2n number of chromosome which produce also 2c amount of chromosome. 2c represent, it's a c value paradox which represent the amount of chromosome that is present in a nucleus or cell. But in the case of mitosis, there is also a diploid cell undergo meiosis. The two condition is 2n and 2c. Then after meiotic division 1, they produce two daughter cells. They are haploid in nature, but the amount of chromosome is 2c. N2c and N2c. Then meiosis 2, which leads to the formation of 4 daughter cells. They are haploid in nature and the amount of chromosome is 1c. So NC, NC, NC. The N which represent the number of chromosome, C represent the amount of chromosome. That means after the meiotic division or reduction division, the chromosome number reduces and also amount of DNA which is reduces by half. But in mitotic division, the number of chromosome and amount of DNA which retain or which remain as the parent. So first mitotic division is known as sequestration division. Meiotic division which is known as reduction division in our question. But the question is, the enzyme recombinase is required at which stages of meiosis 1? Options are Kcet, zygote, diplotin and diakinesis. Kcet is a stage where the recombinase enzymes are formed. Zygote, synapses occur. Diplotin, X-shaped chiasma formation takes place. Diakinesis, terminalization of chiasma. So the answer is Kcet. Okay, then move to the next question. Oh, it's a statement question. Homeostasis is a condition where the organism, homeostasis, it's coming from organism and population. The chapter, second year chapter, organism and population. In organism and population, we study the organism. They are coming under a particular group which is known as regulators. Regulators and conformers. Regulators have the ability to change their internal environment according to the external environment. If the temperature, outside temperature is increases, the internal temperature also, these organisms, this regulator change their internal temperature also or increases according to outer temperature. If the outer temperature is decreases, this organism also decreases their own temperature. That means the particular group which regulate their temperature or their salinity according to their outer environment which is coming under a regulator. If an organism has no this type of efficiency, they are coming under conformers. Many of the organisms, they are act as regulators. These regulators generally are of thermoregulators. Thermoregulators. These thermoregulators, the term thermoregulation, which means this organism having the ability to regulate their inside or internal temperature according to the outside temperature. So thermoregulator actually doing a homeostasis effect. Homeostasis is a condition where, and the options are, maintain a constant internal environment in ever-changing external environment. Then do not maintain a constant internal environment, change their internal environment according to their external environment, change their internal environment when the external environment is constant. Here the answer is option C, change their internal environment according to their external environment. That process is known as homeostasis. and that organism coming under the category that is known as regulators. In this chapter, organism and population, we study the characteristics of the organisms, their regulation, then conformation, etc. So, these are all about homeostasis.